Assalamu alaikum warahmatullahi ta'ala wabarakatuh. I'm Salma al-Mahdi, and today we are going to discuss metabolism of purine nucleotides. All forms of life, with the exception of protozoa, can synthesize purines and pyrimidines from amphipolic intermediates. So we as humans, when we take dietary purines and pyrimidines, we are not going to directly incorporate them into our tissues. When we want to make our own DNA and RNA, we will use amphipolic intermediates to synthesize our purines and pyrimidines. We are not using ready-made purines and pyrimidines in the diet. Thus, purines and pyrimidines are dietary non-essential because we can't make them. Our objectives are to identify the requirements for purine biosynthesis, to describe the layout of purine biosynthesis, to describe the role of 5-phosphoribosyl 1-pyrophosphate as an intermediary metabolite, to describe the role of inosine monophosphate as a parent intermediate for both adenosine monophosphate and guanosine monophosphate, to define and identify the role of what is called salvage pathway, to describe regulation of purine biosynthesis, describe synthesis of purine deoxynucleotide, list example of drugs that inhibit purine biosynthesis and catabolism, list examples of relevant disorders. The requirements for purine biosynthesis include 3 amino acids: aspartate, glutamine, glycine, tetrahydrofolate, ATP, and enzymes. As you can see here, glycine, the whole molecule of glycine, the 2 carbons and the 1 nitrogen is incorporated into the purine molecule, making up carbon atoms 4 and 5 and N7. N1 comes from aspartate, N3 and N9, both of them come from the amide nitrogen of glutamine. Tetrahydrofolate provides carbon atom number 2 from N10 formyl tetrahydrofolate and carbon atom number 8 from N5 N10 methenyl tetrahydrofolate. Regarding the layout of purine biosynthesis, this molecule here, INB, inosine monophosphate, is the parent molecule for synthesis of both AMB, adenosine monophosphate, GNB, guanosine monophosphate. So when the body wants to synthesize purines, we will start by amphipolic intermediates, here represented by the ribose 5-phosphate in addition to ATP. It will proceed through 11 reactions to form INB, and from then the reaction of the pathway bifurcates into that of AMB or that of GNB. The first step is synthesis of a very important intermediate called BRBB, which stands for 5-phosphoribosyl 1-pyrophosphate. BRBB is important because it is an intermediate in purine biosynthesis, in purine salvage pathway, in the biosynthesis of NAD and NADP, and in pyrimidine biosynthesis. BRBB has also an additional importance that will come across it as we go through the lecture. So the first reaction is synthesis of BRBB by BRBB synthase. We start by ribose 5-phosphate. We use ATP as a source of pyrophosphate, and these are pyrophosphate added here. So it is a 5-phosphoribosyl 1-pyrophosphate because the pyrophosphate is attached to carbon atom number 1. The second step is transfer of amide nitrogen of glutamine. As you know, glutamine amino acid has 2 nitrogens, one amino group and one is an amide nitrogen. So when it is amino nitrogen, is going to be donated to the BRBB by a BRBB glutamate aminotransferase. This is glutamine, the nitrogen donor. We need water. There is hydrolysis or release of pyrophosphate and glutamate. And this is the amide nitrogen now of glutamine. And we have the compound 5-phosphodieribosylamine. Third reaction is a condensation reaction whereby 5-phosphoribosylamine condenses with the whole molecule of glycine to form glycinamide ribosyl 5-phosphate. ATP here is needed. The fourth reaction is a formylation reaction whereby carbonate of inosine monophosphate is derived from N5N10 methanol tetrahydrofolate by a formyl transferase. We have formyl glycinamide riboside 5-phosphate. And then we have a transfer of an amine nitrogen from the second glutamine. So for each purine molecule, we need 2 glutamine amino acid. Each one of them will donate disamide nitrogen to form formyl glycinamide riboside 5-phosphate. The sixth reaction is dehydration and ring closure, forming amino imidazole riboside 5-phosphate. This is the first ring, and you know purine has 2 rings, so now the first ring is complete by synthetase. This dehydration. The seventh reaction is a carboxylation reaction, and this carboxylase here does not require biotin and does not require ATP. Amino imidazole carboxylate riboside 5-phosphate is formed. The eighth reaction is a condensation reaction with aspartate by synthetase, forming amino imidazole succinyl carboxamide riboside 5-phosphate. The whole molecule of aspartate is incorporated into the molecule with the release of water. And then we have libration of the carbon skeleton as fumarate by adenosyl succinase. So fumarate is released, and now the second ring is about to be formed. And we have amino imidazole carboxamide riboside 5-phosphate, and then we have formylation where the source would be N10 formyl tetrahydrofolate by a formyl transferase to form formido imidazole carboxamide riboside 5-phosphate. In the 11th reaction, we have ring closure by a cyclohydrolase with release of water and inosine monophosphate, and B is now formed. From here, we can synthesize either AMP or GMP. Regarding formation of adenosine monophosphate, we start by addition of aspartate to I and B to form adenosyl succinate, and then by synthetase, there is a release of fumarate, just similar to the other reaction I've just come across. So inosine monophosphate with addition of in this reaction here, is similar to this one here with release of fumarate. So for formation of A and B, we have addition of aspartate to inosine monophosphate to form adenylosuccinate, AMBS. And then by adenylosuccinase, there is the release of the carbon skeleton and formation of adenosine monophosphate as we know. Regarding formation of guanosine monophosphate, there is first oxidation of inosine monophosphate to the inosine monophosphate by a dehydrogenase, followed by transamidation. A and B by a dehydrogenase will give us xanthosine monophosphate, XNB. Glutamine here donates its amide nitrogen, so it is the enzyme here is a transamidase. It is not transaminase because the transfer active group here is an amide group and not an amino group to get guanosine monophosphate. So if you want to have nucleoside diphosphates or nucleoside triphosphate, you just employ the action of kinases. For example, from GMB to GDB by a kinase, from GDB to GTP to guanosine triphosphate by another kinase. ATP can either be formed from ADP by oxidative phosphorylation, as you know, or phosphorylation at substrate level. We have certain drugs that inhibit purine synthesis. Examples include antifolate drugs, which is a group of drugs that inhibit synthesis of purines, particularly as enzymes that require folate, the two steps that require folate. Examples include 6-mercaptopurine, which is used in the treatment of leukemia and inflammatory bowel disease, and mycophenolic acid, which is used as an immunosuppressant in post-transplantation reaction as well as many rheumatic conditions. Glutamine analog, such as azacitidine, is used as an antineoplastic and an antibiotic. Diazo-norleucine inhibit reaction number 6 of purine biosynthesis. Now we come to what is called salvage pathway. When you salvage something, you preserve most of it. You do a beneficial overall effect economically. This is what we mean here in regard to purine biosynthesis. Salvage pathways are reactions that convert purines and their nucleotides to mononucleotides, and they require less energy than the de novo synthesis. So, if the body needs a mononucleotide or a nucleoside, it doesn't have to start from the anabolic intermediates from the very beginning, then going through the 11 reactions to reach INB and then to synthesize ANB or GNB. No, it will use a ready-made or an already present nucleoside to have to form a nucleotide, or to use a ready-made purine and to add the sugar to it to form a nucleoside. Liver is a major site for salvage purine biosynthesis. Liver provides purines and their nucleosides for tissues that are incapable of synthesis, such as the brain, red blood cells, polymorphonuclear leukocytes. For purine salvage pathways, there are two mechanisms: either phosphoribosylation of a free purine by PRPP as a source of phosphate ribose moiety. Quantitatively, this type of salvage reaction is the most important one. It will form a purine 5 mononucleotide. So the phosphate group will be at position 5' of the sugar. So this is our base. The purine is already there. So instead of synthesize it from the very beginning, from the scratch, no. We are going to use a ready-made purine. We just add to it a ribose and phosphate moiety to form phosphoribosyl purine. And this reaction is by adenine phosphoribosyl transferase. If we want to form an adenine monophosphate, and ribosyl guanine phosphoribosyl transferase if we want to synthesize GNP, guanine monophosphate. So adenine is adenine phosphoribosyl transferase. Phosphoribose moiety will be added to form ANP. And this is hypoxanthine guanine phosphoribosyl transferase, catalyzing for conversion of hypoxanthine inosine monophosphate and guanine to guanosine monophosphate. Hypoxanthine guanine phosphoribosyl transferase, abbreviated as HGB R for riboside T. HGBRT enzyme is very important for a classic technique that is called, or technology that is called hybridoma technology, which is very important or have revolutionized the production of monoclonal antibodies. Go and read about it, and and then you can appreciate the central role of this enzyme for synthesis of hybridomas and production of monoclonal antibodies. The second reaction for salvage pathways is phosphorylation of a purine hyponucleoside. In the previous reaction, we started with a base and we add to it the phosphoribose. Here we have the nucleoside, so we have the base, the purine, plus the ribose. We just need the phosphate, which comes from ATP. So adenosine will be converted to adenosine monophosphate by a kinase. Deoxyguanosine will be converted to deoxyguanosine monophosphate by deoxycytidine kinase. How is purine biosynthesis regulated? Hepatic purine nucleotides biosynthesis is stringently regulated. The PRPP concentration determines the overall rate, and this is what I have mentioned earlier when I told you that PRPP is a very important molecule. In addition to it is being part of already listed 4 metabolic pathways, it has a regulatory role. Its concentration determines overall rate of purine biosynthesis. Rate of synthesis of PRPP itself depends on the availability of ribose 5-phosphate, the substrate, and activity of its synthesizing enzyme, the PRPP synthase. This enzyme is sensitive to phosphate concentration and to allosteric regulators such as purine nucleotides. There is feedback inhibition of PRPP glutamide amidinotransferase, which represents the first committed enzyme in purine biosynthesis. There is also feedback inhibition by both AMP and GMP because they are the final products. They are competitive inhibitors of PRPP. However, less significant than regulation by PRPP synthase in the first type of regulation. This is the pathway, and this is to show you how the products have negative feedback on the earlier pathways. But significantly, or the most significant regulator here is PRPP synthase. Less, or number 2 is PRPP glutamyl amide transferase. Feedback inhibition of conversion of IMB to AMB and GMB is a bifurcation at the parent molecule to the two pathways. AMB inhibits adenosine succinate synthase and GMB inhibits IMB dehydrogenase. There is cross-regulation between the pathways of IMB metabolism, which decreases synthesis of one nucleotide when the other is deficient. That means they go hand in hand. So GTP here can activate this pathway, and this ATP here can activate this pathway. This will ensure that there is almost equal molar concentration or production of two nucleotides at the same time, because if you want to synthesize a nucleic acid, you need A for G and G for A, so they must be present at equal concentration to be used as building blocks for nucleic acids. So far we have talked about ribose. What about deoxyribose in the deoxynucleotides? That's needed, for course, for the DNA synthesis. Deoxynucleotide in purine biosynthesis is by the reduction of purine at carbon atom number 2 of the sugar 2' by ribonucleotide reductase complex, which forms deoxyribonucleotides, diphosphates, DNPs. The enzyme is active only when the cell is synthesizing DNA prior to cell division. So otherwise, in the rest of the cell cycle, this enzyme will be dormant or not active. And this enzyme is subject to complex regulation, which ensures balanced production. NADPH here is the source of hydrogen for the reduction, and this is our complex enzyme that reduces ribonucleoside diphosphate to 2-deoxyribonucleoside diphosphate. So far, we have discussed purine synthesis. What about purine metabolism? How the body breaks down purines. Humans catabolize purines to uric acid. Some mammals possess a uricase enzyme which cleaves uric acid into allantoin. But we as humans, we don't have a uricase, so our end catabolite, or the end product of purine catabolism, is uric acid. And it is an acid, and the body has to deal with it. Total uric acid excretion in a normal human is around 400 to 600 milligrams per 24 hours. The body needs to maintain uric acid within normal range, both in serum and in urine. Why in serum? Because if the concentration exceeds the normal, it will have the tendency to precipitate in tissues. Many compounds influence renal absorption and secretion of sodium urate, the salts of this uric acid. For example, aspirin competitively inhibit urate reabsorption and excretion. So, to break down adenosine, it is first deaminated to inosine by adenosine deaminase, and then there is phosphorylated hydrolysis with use of phosphate, by phosphorylases of the glycosylated bond of inosine by purine nucleoside phosphorylase to yield hypoxanthine. Hypoxanthine is converted to xanthine by xanthine oxidase, and then xanthine itself by xanthine oxidase again is oxidized to uric acid. So adenosine to inosine to hypoxanthine to xanthine and uric acid. Xanthine oxidase, as you can see here, is catalyzing the last two steps of uric acid formation. It is important because xanthine oxidase is a site of drug action. Regarding guanosine, will be converted to guanine by phosphorylases and removing the ribose moiety, and then by guanase, the amide nitrogen will be removed from xanthine, and then again by xanthine oxidase, uric acid is formed. So xanthine oxidase is shared between adenosine and guanosine catabolism. Uric acid is a weak acid, and at physiologic pH, it exists as undissociated acid, so it will be an acid plus it is conjugate base, which is sodium urate. It is urates in general, but because sodium is the main intracellular cation, it will be present as sodium urate. Sodium urates are more water soluble than uric acid. The more acidity, the less dissociation. In our system, urine acidification occurs at the distal collecting ducts and the distal tubules and collecting ducts. So here, it is most likely there is more acidity, less dissociation. It will be more likely to be present as uric acid, while here it will be more likely to be present as urate. So precipitation, crystallization is likely to occur distally in the tubules. Thus, proximally, urinary tract crystals exist as sodium urates, but distally they exist as uric acid. And since most of the urinary stones are composed of uric acid, stone formation can be reduced by alkalization of urine, for example, by using potassium citrate. And here, as we said, aspirin, because it increases the acidity, will increase the likelihood of formation of urinary tract crystals. When the level of uric acid is high, it is called in serum, it is called hyperuricemia. So hyperuricemia is serum urate level that exceeds the solubility limits, the solubility into urate, sodium urate salts. And what causes hyperuricemia? Generally or broadly, you can talk about abnormal urine excretion. So excretion through the kidneys is abnormal, so the uric acid is retained in the body, or there is overproduction of purines, and then they will be catabolized, leading to an excess amount of it is catabolized the uric acid. Conditions such as malignancy, cancer, because there is unregulated division of cells, and cells will also undergo apoptosis, or they will be targeted, for example, by chemotherapeutics, or they will just die because they are rapidly growing and they require glucose, and when they are deprived of glucose and other nutrients, then plastic cells are likely to die. This will lead to a catabolic state, including catabolism of urics. So in cancer condition and malignant conditions in general, hyperuricemia is expected. Psoriasis is an inflammatory skin condition in which also there is increased division of the outer layers of the dermis of the skin. So the skin, the keratinocytes will divide at an abnormal rate, forming plaques, which is called plaque. We have different subtypes of psoriasis, but generally the concept is similar to cancer, in which there is more than normal or higher than normal division of cells, and Consequently, there is higher production of or increased production of purines and their catabolite uric acid. Enzymatic defects can also lead, of course, to overproduction of uric acid. And on the other hand, we have hyperuricemia, which is below normal levels of uric acid, which results from oxidase deficiency, um, leading to reduced uric acid level, hypoxanthine level, xanthine, and dancing lithiasis. That is stones formed from xanthine in urine. Regarding disorders, enzymatic disorders, gout, the hyperuricemia, can result from deficiency of PAPP synthase or HDPRT, hydroxyzine guanine phosphorylcysteine transferase. It is characterized by purine overproduction and overexcretion. It is X-linked condition. Leishman syndrome is a deficiency of HDPRT, X-linked, and also lead to purine overproduction and overexcretion in addition to self-mutilation. Patients who have this genetic condition injure themselves, bite themselves, and render self-harm, which is peculiar about this syndrome. The deficiency in the form of ADA deficiency and BMP deficiency, adenosine deaminase and purine nucleoside phosphorylase. These enzymes are needed for both T cell and B cell. Adenosine deaminase, if it is deficient, it will result in a combined T and B cell deficiency plus adenosinuria. That is presence of adenosine in high concentration in urine. A combined T and B cell defect is called SCID, severe combined immunodeficiency. It is a serious condition because your two major cells of the adaptive immunity, the T and B cell, are deficient. It occurs in autosomal recessive pattern. PMB will lead also to T cell deficiency, and because B cell depend on T cell help, we expect also a B cell abnormality in function, at least if not in quantity. Renal lithiasis or renal stones result from adenine phosphorylcysteine transferase deficiency, an enzyme of Sabel pathway. It is autosomal recessive inheritance. Xanthinuria from xanthine oxidase deficiency lead to renal lithiasis and hyperuricemia. It is also autosomal recessive. Gout is famously known as a type of arthritis, that is gouty arthritis. It results from hyperuricemia. Your serum level exceeds solubility limit. This will lead to crystallization and deposition in soft tissues, causing inflammation. It can present acutely as acute gouty arthritis, which is a very painful condition, and it may be a chronic condition progressing to what is called chronic gouty arthritis, which is also called tophaceous gout. Sodium urates appear as needle-shaped negative birefringent crystals in synovial fluids. So for diagnosis, we can aspirate via needle. We can take part of the synovial fluid. Fluids lubricates inside of the joint, and when we put it under a special type of microscope with plain polarized light, if the patient has gout, hyperuricemia, red crystals will appear like this. This is called sodium birefringent, and it is characteristic of gout. And this is a picture of an inflamed first toe joint, which is classically associated with gout. As I told you, it is one of the very painful conditions. And this is an acute, because you can see here it is red, it is swollen, so it has the signs of acute inflammation. And this one here, this is swelling here is result from crystal uric acid crystal deposition in addition to inflammatory tissue, and this is a chronic form of gout, or what is called tophus. This sign here, this is a tophus, and the whole condition is called tophaceous gout. This does not resolve unless it is surgically removed. It means that this patient has gout for so long and it is not well controlled. And of course, there are drugs that control the level of uric acid in our blood, and in fact, gout needs to be managed seriously with the same seriousness of managing diabetes and hypertension, because all of them are part of what is called the metabolic syndrome. This is all. Thank you very much.